is from Associate Professor Tina Seelos, and who's going to talk about the investigation of activating autophagy to treat Alzheimer's disease. Tina has had over 28 years of working in healthcare, biotechnology, academic research, clinical research and management sectors in senior clinical roles. She's passionate about mentoring future industry leaders and advocating for the Australian R&D community. And Tina serves on several therapeutics executive advisory boards. So I'd like to welcome Tina to do her presentation for Seelos Pharmaceuticals.
Hi everyone, hopefully you can all hear me. And I just want to say it's so nice to be back here after 3 years and see everyone face to face. Lots of old faces that I've known and worked with over the past 10, 15 years and lots of new faces. I also, before I start, I want to say congratulations because I don't think you all understand how important and how amazing you all are and what you've actually achieved. First of all, kudos and congratulations to all the big companies in this room today for the investment, the knowledge, the resource, the money that's gone into and commitment to developing, something for a cure or treatment for a disease that's one of the most significant and impacting in our generation and generations to come. Also, of course, that the congratulation goes to the sites because these are some of the most complex site and patient burdening studies you will ever be involved in. And I can vouch for that because in my new life under Alithia Life Sciences, Mary and my team and I look after a portfolio of companies, one of which is Seelos, I'm going to talk about today. But we look after all different indications, and I can tell you Alzheimer's disease clinical trials are one of the most intensive in terms of patient and site burden. So just take a minute and think about that before I start. But today, as I said, I'm talking about Seelos, and I also want to talk about what they're doing in Australia and also globally, and just a little bit of reflection based on the talk this morning by you, Michael, about COVID and the impact. For me personally, one of the impacts of working from home the past 2.5 years, not directly because of COVID, but I guess indirectly because I've been working from home, is that in my mind, and because I haven't been patient-facing day to day and just working away on my computer, 16, 18-hour days, I don't usually or often pronounce some of the words that I work with. And a good indication of that is today I'm talking about autophagy, but in my mind I've been pronouncing it from the Greek word autophagia. So in my mind I've had to retrain my brain to actually say things out loud. So today, talking about a company that is very dear to us because I've been working with them for nearly 12 months now. And they are a company that have a head office, a small team. And the other thing I was going to say was the answer may not be from the big pharma guys, and no offence to that, no disrespect intended. And it's really beautiful to see a good representation from the smaller guys, one of which is Seelos Therapeutics today. Because until we look, we will never know. So today I'm going to talk about one of their small proof of concept studies they're doing here in Australia. So Seelos, a small, agile team based in the US, in New York, is their head office. Most of our US colleagues are based throughout East Coast and some in Seattle. Mary and I are project leads for Seelos based here in Melbourne. So we look after their studies. So think of us as their project leads, the people that interface between the CRO and the sites for these studies. So Seelos is doing various different things as depicted in this slide, some of which are involved trials in Australia, some are not. The first one on the left-hand side is SLS002 program, which is actually has fast-track designation because it's such a huge unmet need. And they're trying to get a wafer, which is a ketamine wafer, for treatment in acute suicidal ideation behaviour and major depressive disorder. Through COVID, they completed their phase 1 study and they then successfully have started their phase 2. The first part of that phase 2 and the second part is ongoing at the moment. And really, because it's fast track, they're hoping to use that data to register that product. On the far right-hand side, they're doing the SLS 004 and SLS 007 projects, and they're in Parkinson's disease, so still CNS. But in this case, they're gene therapy products. The 004 is looking at targeting the regulation of SNCA, the alpha-synuclein gene. And the 007 is actually still a gene therapy program, but that's targeting the aggregation of alpha-synuclein. But today I'm going to focus on the middle box, and that's the SLS 005 program, which is using a product called Seelos, and I'll talk about that in a moment. They've got some various diseases in the CNS space that they're addressing with Seelos. One of those is ALS, and they've successfully been able to get a really prestigious and large grant as part of the Healy Initiative, and they're fast recruiting, almost done with recruitment for a global ALS trial. The second is a fast track designation based on encouraging phase 2 open label results in spinocerebellar ataxia, SCA. And again, they're just about to launch, and in fact they have launched a global phase 3 study looking at one of the SCAR subtypes as well that they're hoping to bring to market. And today I'm going to talk about, they're also doing a study called the Basket Study in Australia, which is looking at Huntington's as well as ALS and SCAR. Okay. And I'll talk about the other study that we're launching in Australia at the moment. So Seelos is, unlike a lot of the more complex, I guess, investigational products this room is used to addressing and being involved in, this is relatively simple in terms of what it is. Seelos is a simple disaccharide composed of 2 glucose molecules. It's found extensively in nature and food products. We as humans can't synthesize it, but we can metabolize it. We know that Seelos is found in the gut and liver and kidneys, but even though it's found in the gut and the Seelos enzyme is in the gut, we can't metabolize it in the gut. So therefore, the investigational product in our trial of Alzheimer's disease patients will use an IV infusion of Seelos. So the rationale, there's lots of preclinical evidence in the literature, which is what they have based the current study on. The rationale is that Seelos is known for its protein stabilizing properties and its ability to activate autophagy. And we know that these pharmacological mechanisms sort of really fit nicely with the impact of beta amyloid and tau in the development of Alzheimer's disease. Okay. So I'm not confessing to be an expert in this diagram here, but I can tell you that based on the literature that's been pulled together as a basis for the current project, we know that Seelos significantly decreases both amyloid and tau, and we know that it does this through increased lysosome biogenesis, through increased autophagocyte formation, and of course then through molecular chaperone activity as well. So that we know is the mechanism of action, which people always like to know and ask. So if we look at a little summary of the preclinical information that's available for Seelos in animal models, they are both in mouse and rats. In particular, when Seelos has been given as an intracerebral injection in mice, it's been found that beta amyloid deposits have been significantly reduced in the hippocampus of the mice, and in terms of cognitive behaviour using the Morris water maze test, they've been improved cognition has been noted in these animals as well when they've been treated with Seelos. But even if we give mice Seelos orally via gavage, we see nice increased synaptic activity, and that's also again via increased cognition observed by the Morris water maze test. But also importantly, and I know that it's a huge area, the biomarkers, that then a surrogate biomarkers, I should say, of activity of improvement in these type of conditions. There's evidence to show that when these animals are given Seelos, that biomarkers such as synaptophysin and progranulin have also have been increased in the hippocampus of these animals as well, and also the cortex as well. And the next few slides just show this data pictorially. So on the left-hand side, you can see this is in vitro. So these type of cells are an Alzheimer's disease cell type, and you can see that when you treat these cells with Trehalose, that you decrease the amount of both beta amyloid 40 and beta amyloid 42. And also on the right-hand side, when you have transgenic mice, which an Alzheimer's disease transgenic mice model. In this case, it's the amyloid precursor protein, transgenic mice. You can see that if you treat them again with Trehalose, that you decrease the generation and the formation of plaques in these brains. The next one as well is with regards to tau. And when we look at tau transgenic mice and treat them with Trehalose, you can see again that the aggregates in these, the number of aggregates are significantly decreased just with the treatment of Trehalose. And lastly, just with the Trehalose, the cognitive performance, again in the APP transgenic mice, you can see particularly in the left-hand side where we see the Morris water maze test that the Trehalose treatment and the wild type are very, very similar. Showing the effect of Trehalose on these mice and on cognition, I should say. And, I thought that was last, but this is the last preclinical slide, and this is just showing a different model of transgenic mice. This time is TG2576, and these mice again treated with Trehalose, and you can see that both in the hippocampus and cortex, there's a significant increase of these surrogate biomarkers such as synaptophysin, also double cortin and progranulin as well. So lots of evidence of preclinical information to base our clinical study on. And just the next few minutes, I just want to make a note of the study that we now have up and running in Australia. It's only going to be done in Australia. At the moment, we have 3 sites. 1 in Western Australia and 2 in Victoria. And I'm standing up here unashamedly saying that if you're interested in the study, want to take part, we do need a few more sites to make this go a lot quicker. So please either contact myself or Mary. Be happy to talk to you about the project. So it is looking at 40 eligible participants, and they'll be randomised. There's about a six-week screening period, and if they pass all of the screening based mainly on a CSF test to substantiate that there's presence of beta amyloid as well, then they'll be randomised in a 3-to-1 ratio, either Trehalose or placebo. This project is a little bit different because it does involve weekly infusions of Trehalose. And that is a burden to the sites, we get that, but there is a very generous budget, and also we've thought about home visits to take the burden off the site as well. So we've got pretty much ready to go, all of it's in place. We need patients, we need a few more sites, and they'll be followed, they'll be treated for 52 weeks. There is hope that at the end of the 52 weeks we'll expand to an open label extension. But yes, hopefully we're off and running with the project, and talk to us if you're interested. But thank you for your time.
Thanks, Tina. Any questions at all? No. No, it's got the mic. Okay. Thanks, Tina. And exudes, Tina, so thanks very much. Yeah. No, I think that I second your comments.